everyone, I'm Priyanka and today we will talk about scanning electron microscopy. Scanning electron microscopy, also abbreviated as SEM. Electron microscopy use electron for imaging in a similar way that light microscopes use visible light. Scanning electron microscopy uses a specific set of coils to scan the beam in the raster-like pattern and use the electrons that are reflected or knocked off the near surface region of a sample to form an image. Since the wavelength of the electron is much smaller than the wavelength of the light, the resolution of scanning electron microscopy is superior to that of the light microscopy. There are two main types of electron microscopy: transmission electron microscopy, which detect electron that passes through a thin, a very thin specimen. Second, we have scanning electron microscopy, which uses the electron that are reflected or knocked off the near surface region of the sample to create an image. Talking about the scanning electron microscopy working, as you can see the systematic representation of the basic scanning electron microscopic components in figure 1. In this type of electron microscopy, the electron beam scans the sample in a raster pattern, but first electron is generated at the top of the column by electron source. These are emitted when their thermal energy overcome the work function of the source material. They are then accelerated and attached by the attracted by the positively charged anode. As you can see in the figure, first we have a electron source, then an anode, then we have the condensing lens, then scan coils, and then the objective lens, and then finally the beam will fall on the sample, and further the image will be seen by a secondary electron detector. The entire electron column needed to be under vacuum, like all the components of an electron microscope. The electron source is sealed inside a special chamber in order to preserve vacuum and protect it against contamination, vibration, or noise. Although vacuum protects the electron source from being contaminated, it also allows the user to acquire a high resolution image. In the absence of vacuum, other atoms and molecules can be present in the column. Their interaction with electron cause the electron beam to effect reduce the image quality. Furthermore, high vacuum increase the collection efficiency of electron by the detector that are in the column. In a similar way to optical microscope, lenses are used to control the path of the electron. Because electron cannot pass through glass, the lenses that are used here are electromagnetic. They simply consist of a coil of wire inside metal pole pieces. Uh, when current passes through the coil, a magnetic field is generated. And electrons are very sensitive to magnetic field. Their path inside the microscope column can be controlled by these electromagnetic lenses simply by adjusting the current that is applied to them. Generally, two types of electromagnetic lenses are used. The condensing lens is the first lens that electron meets as they travel towards the sample. This lens converges the beam before electron beam cone cone opens again and is converted again once more by objective lens before hitting the sample. The condenser lens defines the size of the electron beam, which defines the resolution, while the main role of the objective lens is to focus the beam onto the sample. The scanning electron microscope lens system also contains the scanning coils, which are used to rest the beam on the sample. In many cases, apertures are combined with the lenses in order to control the size of the beam. The main component of the typical scanning electron microscopy are shown in the figure 1. Now talking about the formation of the image, the interaction of the electron with the sample can result in the generation of many different types of electrons, ions, photons or irradiation. In case of scanning electron microscopy, two types of electron used for imaging are the backscattered and secondary electron. Backscattered electrons belong to the primary electron beam and are reflected back after elastic interaction between the beam and the sample. On the other hand, secondary electron originated from the atom of the sample. They are result of inelastic interaction between the electron beam and the sample. Backscattered electrons come from the deeper region of the sample, as you can see in the figure 2, while the secondary electron originate from the surface region. Therefore, backscattered electron and secondary electron carry different type of information. Backscattered electron images show high sensitivity to difference in atomic number. The higher the atomic number, the brighter the material appear in the image. Secondary electron imaging can provide more detailed surface information. In many microscopes, detection of the X-ray, which are generated from the electron-matter interaction, is also widely used to perform metallic analysis of the sample. Every metal produces X-ray that have a specific energy. The X-ray are the material fingerprint. So by detecting the energy of the X-ray that come out of the sample with the unknown composition, it is possible to identify all the different elements that is contained. That's all we have in scanning electron microscopy. Thank you and have a nice day.